Okay, moving along, we just spent last time talking about soil testing in the context of evaluating fertility. And here we're going to be talking about not soil testing, but the other broad category, which would be plant testing. And this kind of encompasses a wider range of different offerings in terms of the approaches and the methodologies for testing. So we'll go through these kind of one by one and talk a little bit about. So let's just start before we get into it, some about plant nutrient deficiency symptoms, okay? So nutrient deficiency appears when a plant simply cannot function properly given the nutrients it has. We've talked about the essential nutrients that plants have. Nutrient deficiency symptoms appear during early growth and may disappear after growing season progresses. This is very common in Ohio when soils might be very cool or overly wet or overly dry in the springtime. That root system in a new crop has not fully developed, hasn't explored kind of the soil profile. And for a variety of biotic and abiotic reasons, it's not, they're having problems getting all of the essential nutrients that they need at that point in time, okay? Visual symptoms should only be used to support soil or plant analysis. So when we're talking about, and we'll move into this in a second, but visual symptoms, it's really our first line of defense or our first clue that something's wrong with that plant. That plant's not happy, it looks sick, it looks unhealthy. Really, there's a lot to be said about the power of that observation, but we always tell people that that is not we shouldn't look at that as a definitive diagnosis, but so we should use that as a guide and help figure out if we need to take soil samples and plant analysis, and then using those results from the lab to actually help fully diagnose and validate kind of what we initially suspected. So recommendations based on nutrient symptoms really don't have, recommendations are based on the soil test values and the plant analysis that we get back. And so just thinking about the nutrient deficiency and nutrient symptoms, okay? Again, here's this kind of classic curve that we see where on the x-axis we have concentration of nutrient in tissue, okay? So that we send this to a lab, they provide essentially a concentration. On the y-axis, of course, is the percent growth as a percent of maximum. We could think of this as relative yield or just overall productivity in some kind of general sense. When we hit below, of course, a critical nutrient range or a critical level, that plant is going to be driven into nutrient deficiency. Above that level, we're good. Depending on the nutrient the plant is able to luxury consume, that is store nutrients, in excess of what they actually need. And then at some point we move far enough in the concentration where toxicity becomes an issue. Okay, too much of a good thing. Again, we talked about soil testing last time. Here we're talking about plant testing. And so really we can break these down into three different categories: visual symptoms, plant analysis, and plant sensing. And we're going to talk about the visual symptoms right now. So symptomatically, there's a lot of things that can go wrong with plants, and we can characterize them by chlorosis or a yellowing of tissue, or even necrosis, which is the death of tissue. There can be stunting, there can be kind of curling of the leaves or just abnormal growth. There can be a lot of different things that might go on. We can talk about these visual symptoms in the context of the spatial orientation or the how widespread it is on the leaves. So, we can have a normal leaf here. We can have a localized chlorosis, which might be just the leaf tip or the base of the leaf, or where say the petiole might attach or what have you. It can be generalized, which means across the entire leaf. We can think about things where there's necrotic spots, or there's intervenal chlorosis. You'll hear this. So essentially, here the veins of the leaf, and then not the vein itself, but within that, in between that venal tissue, intervenal chlorosis. It's actually yellowing. The chlorosis of the veins would be the opposite of that, where the veins of the leaves are actually the ones that are experiencing problems. And there's quite a bit of other possibilities. Just a purple pigment that we might see, which might be stress response from anthocyanin accumulation. This is what we see, for example, with phosphorus deficiency quite a bit. Okay. So a word of caution that visual symptoms can come from many possible sources. So it can be not just the nutrient that you are thinking of, but it might be, a combination of nutrients. It might not have anything to do with nutrient deficiency. It might have something to do with damage from insecticides, herbicides, actual insect predation, or disease. It's very common for, when we're kind of as a extension team diagnosing issues that people are experiencing in the field, that we will send emails back and forth with photos or text messages showing, this kind of chlorosis and necrosis, and some of the comments might be, well, it doesn't really look like a nutrient deficiency. I think it's probably a disease, or vice versa. This isn't a disease. I think it's nutrient related. So, there can be certainly symptoms that look very similar and can be easily confused, right? Soil conditions, again, these would be like more abiotic. So things like the temperature, the moisture, content of the soil. If that soil is really compacted, or even if it's not compacted, if it was planted wet, and say we have some sidewall compaction, so the roots haven't fully developed, or haven't had a chance to kind of branch laterally out of the rows, that's an example. Salinity, pH. Of course, there's nutrient interactions, and we'll talk more about these as we get into it, but an over-application of P can induce zinc deficiency, because these two nutrients compete for uptake into plants. And toxicity problems, again, kind of thinking about what one nutrient there might be an excess of, and understanding that it's not, say, a nutrient deficiency, but it's actually toxicity. And that could be from starter fertilizer, or some kind of foliar application at too high rates, not following manufacturer specifications. These things happen as well, of course. Okay, so again, just because you see something wrong with a plant, it behooves us to understand that it's not automatically some nutrient deficiency, right? It could be a suite of possible things. Okay, so when we're talking about kind of this first line of defense, which is just our visual observation, and thinking about trying to kind of pinpoint, there's a really nice way that we can, in some ways, eliminate a lot of possibilities. Okay, so it's like essentially this dichotomous tree. And we talk about nutrient mobility in plants. So we can describe mobile nutrients and immobile nutrients, and these are not. It's important to understand that we're not talking about mobile nutrients in the soil. We're actually talking about once they get into the plant, are they actually able to translocate within that plant? Or are they basically kind of fix into plant structures and remain there? And so mobile nutrients include nitrogen, phosphorus, potassium, magnesium, chloride, and molybdenum. So what does that mean? It means that when we see deficiencies, visual deficiency symptoms of these nutrients, it typically is going to manifest in older leaves first. So what that means is, if corn or whatever the plant is growing, right, the older leaves have say enough access and they have fully developed, But as that plant continues to grow and continues to pull up these nutrients and say it's driving deficiency, well, then the plant has the ability to essentially capture, um, basically cannibalize itself, immobilize nutrients from older structures, that is lower leaves, and then divert them into those newly formed growing structures and the newly formed leaves, the leaves that are growing, okay? And so, for the three main nutrient constraints in Ohio agricultural systems, N, P, and K, almost always manifest or show up in those lower and older leaves first, okay? Contrary, immobile nutrients, nutrients that are that plants cannot essentially translocate, include sulfur, calcium, boron, iron, manganese, zinc, and copper, okay? So most of the micronutrients, and certainly the micronutrients that we find commonly deficient, which would be things like boron, zinc, manganese, and copper, in Ohio, those are immobile. So the deficiency symptoms are going to show up in those older plants, those younger leaves first, right? So this idea that as the plant grows, maybe the lower, older leaves look fine, but then when we get up that new growth, those younger leaves, that's really where the deficiency symptom is going to manifest, okay? So that's kind of a 1-2, it's either this or that. We can think about this is just a nice chart to show essentially a dichotomous key where you're looking and we've got younger leaves affected first versus older leaves affected first. And again, here's NP and K down here, magnesium, and then your micros, some sulfur and calcium over here. So with some descriptors about what each one might look at. And we'll get into this as we cover each nutrient throughout the semester, right? So we're not going to spend time, but I just wanted to show this. You'll see this multiple times throughout the semester. Here's another way. It's really kind of the same information, but just organized maybe a little bit different. So these might be helpful for, you all to print off or just think about, use this as quick reference when you're in the field, for example. Okay, so that's visual symptoms. Let's move on to plant analysis, okay? So we can categorize plant analysis into either field-based or lab-based. They both, like most things in life, have advantages and disadvantages. When we are in the field, typically kind of doing any sort of plant analysis in the field is fairly inexpensive. You get that information right there the same day, and you can make management decisions really quickly and move on. The disadvantage of field-based measurements is that measurements might not be precise or particularly as accurate as we'd like. Oftentimes we're talking about, say, if we're lucky, a low percentage in terms of nutrient concentration, and many times it's, say, a part per million measurement. So some kind of field-based sensor might not be as accurate as we'd have in the lab. And so the lab is really just the converse of those, right? So the lab, the advantage is it's more precise, accurate. There's greater data quality control. You've got, dedicated technicians and instrumentation, and there's long-term storage of data. There's a lot of nice things that go in that with laboratory testing. Disadvantages, it can be more expensive depending on what you're looking at, and results usually don't get results back for a few days. And so even though the turnaround time is relatively quick, you might want to make a decision about what to do or not to do right then in the field. So there's advantages and disadvantages to both. For agronomic crops, tissue sampling would be in the field. We often do this in kind of what we would characterize as a late vegetative state or an early reproductive state. This is a period of time where plants go through rapid growth, and so sampling here is a good time to really assess as the plants are growing, and then the nutrient uptake rate is kind of at its peak. if we see a deficiency symptom, we're most likely to see it during this time frame. So for corn, that means right at essentially tasseling, or when the silk's coming out and those ears are forming. For soybean, it's typically an upper trifoliate, maybe somewhere around R1 to R3. That's reproductive phases 1 through 3. For a small grain, it would be a flag leaf, which is this last leaf that emerges from right after boot stage and right before that seed head emerges. And then for forage crops, it would be, for example, sampling 6 inches above ground, and then taking that whole biomass and submitting it for analysis right around, say, early bloom or right before bloom for a lot of these clovers and forages. Okay. In-field options, there's a number of different things out there that are kind of used. Cell sap tests are very commonly used in, say, specialty crop industry. They sell things like these nitrate strips, nitrate, and they're like little indicators. You essentially, depending on the system or the crop, but oftentimes you take, say, leaf petioles, and you might crush them, and then you actually measure the sap or the liquid in those, and measure the concentration of the nutrient in that. Okay, there are plant meters that are out there. There's a suite of different things that people might use. We understand that we must sample the correct plant part at the correct sampling time for these things to have kind of validity and provide usable information. A lot of, you'll talk to consultants or growers or people that are in kind of, say, specialty crop. There might be somebody that, like, does this one thing or does that one thing. They're kind of an expert consultant for a crop or a particular industry. And they might have their own system that they've just kind of found and developed and found useful. Well, I really like to take cell sap, right at first bloom, and then again at, 3 weeks later, and you can see what's happening. So you kind of hear these stories that are out there in terms of how people are using these in various different ways, right? So just as an example, we're in a project now that's kind of, we'll be wrapping up at some point, but it's a North Central SARE funded project looking at can we, at hop production in Ohio, Michigan, Indiana, and can we use hop petioles? These are these little stems essentially down here. We can pluck those off, crush those in, say, one of these garlic mincers. Okay, that's a common thing. Collect the liquid in there, and then measure it on this, say, nitrate meter. And so we've given growers these nitrate meters and having them collect data for us and try to track that and link that back with irrigation. So track it over time, say over the growing season, and link it back with what they've irrigated, what they've fertigated, how much they've actually fertilized, and then make some recommendations based on that. Okay. If you're interested in more about this, we've got a playlist here with a few different videos about setting these things up and calibrating it. It's really kind of nitty gritty. For for the hop, we recorded these videos for the hop growers that are participating in this study. Okay. So again, there's guides out there that can look at things and provide ranges. Here we've got, this is from the Midwest Vegetable Guide, where we've got different crops, different stages of that crop, and then different nitrate concentrations or potassium cell sap concentrations. Some of these say ENR, I don't know if that means that there's not good ranges established yet, or that they just don't believe that it's particularly an issue, potassium, say with broccoli or collards, or it's just not a useful guide, for example. So these things kind of exist. When we get into more specialty crops, the information becomes a little bit more scarce and a little bit maybe more industry derived than when we're dealing with like crops that represent large acres or large, you know, kind of economic output for a given state. So a lot of variability that you might actually encounter with some of these things in terms of where the resources are and how vetted or how robust that information is. Okay. Again, we talked about, you know, sampling in corn, and this is, you know, you go around and you essentially find plants, and you might just rip these ear leaves off and collect these ear leaf samples. Do that, say, with 10 plants that look healthy, maybe 10 plants that look a little funky, and then submit those and kind of compare the numbers. For soybeans, we sampled the upper trifoliate, and then we essentially discard that petiole. That's what that is right there. So you just collect the leaves. We analyze those leaves for nutrient content. We talked about flag leaf, etc. And again, you know, depending on the crop and thinking about vegetables here, we might have different guides that provide nutrient concentrations. Okay, so all of these examples for the, you know, the flag leaf, petiole, we would take these, we'd bag these up and either drop them off in a lab or ship them to a lab. The lab is going to dry, grind, and analyze those samples, and then give you a value, and you can compare it to some of these book values that we would call tissue sufficiency ranges. Okay, so we look at a table like this and say, I'm growing carrots. I sampled the fully matured expanded leaves, and our nitrogen concentrations should be somewhere between 1.5 to 2.5% in that. If it's lower than that, we might think of possible deficiency. Above that, we might think that we probably could be in an excess situation. So these published book values are very helpful for understanding the soil test results that you get. Okay. When you submit samples, plant samples for laboratory testing, it's very common procedure in terms of what happens to that sample. They typically will take those, log those samples in, you know, assign the test that's going to be run. They will dry the samples. They will grind them, typically to a 1 mil mesh size. Then they will put this in a heating block, typically very common in the industry, to add nitric acid and then digest them for a couple hours in a heated block here. And that really, once it's ground, and then you put this concentrated nitric acid and essentially digest all of the solid materials into a liquid phase. Okay. Other ways, some people, sometimes they will actually ash, so they'll essentially burn off the sample and have just the ash there. So would be an alternative to kind of the digestion. Then they send it through this incredible machine. So the sample is essentially ground, digested, it's in liquid form. Then they send it through this machine called an ICP. And an ICP stands for inductively coupled plasma spectrometer, okay? And there's different types of these ICPs in terms of what they do and how they actually function. So, and some of these have, you know, things like mass spectrometers attached to the end of them, et cetera, et cetera. So this is complicated stuff, folks, in terms of what it actually does. The take home is it gets, this is an example of an ICP. Almost all of them use argon gas as kind of a torch. It's like you can think of it as a welding torch. It sprays a fine mist of that liquefied sample into the machine. It hits that torch at such a high energy that everything essentially ionizes, right? So all like the proteins or biochemical structures just all become in their atomic form. And that is actually what's read. So when we submit samples and we get, you know, phosphorus or nitrogen, it's not nitrate nitrogen or nitrogen and protein, it's really elemental nitrogen, elemental phosphorus, and these are the values that the lab gives you. Okay, so again, it breaks. This machine will break that plant tissue down into its most basic constituents and read the concentration. So that's really what we're ultimately getting after. If you look at these analysis, this is a percentage base, a ppm base. These are both units of concentration, how much nitrogen per gram or per ounce or per whatever the unit is of of that leaf. And so in the context of here, it's percent nitrogen per per leaf biomass, essentially. Okay. So I've got a YouTube video here. If you want to learn a little bit more about this, I think it's a well done video. In terms of just understanding the complexity, you're not really responsible for it, but it's worth a couple minutes of your time just to kind of come here, skip to about 4 minutes and 30 seconds. It's kind of a sales pitch, but it gives you an idea of how these things operate. It's pretty incredible to think about this. And the reality is these ICPs have, commercial testing labs that are large, even if they're not, they often have 2, 3, 10 of these, and they run all day long, right? And so when you're running soil, you would take soil through these, and they have dedicated ones for plant material as well. We'll talk more about kind of soil testing, getting into that, but again, here, you can just kind of understand these ICPs or inductive coupled plasma spectrometers are the workhorses for most commercial soil testing labs. I mean, they're typically have many of these. Okay. All right. So to wrap up quickly, we're just going to talk a little bit about plant sensing. So we talked about visual symptoms, plant analysis, and plant sensing. So sensing is not an actual true, destructive measurement. It's really a kind of a non-destructive, and that's why we kind of call it sensing. We're not actually like taking leaves and grinding and drying them or crushing them up. It's just seeing the state of the leaf. One of the nice things that are out there is a chlorophyll meter. This is a quick and non-destructive sample. What it really does is, you can see the unit here, you just kind of clip it to a leaf, and it has a light diode, and it just measures the amount of light passing through the leaf and measures the total greenness of that. So it does not indicate like which nutrient is deficient or whatever, but it gives you a value from 0 to 100, and then you can make some inferences based on that. Chlorophyll meters are often related to leaf nitrogen concentrations, and which are often related to grain yield. And so it's, there's a lot of studies out there have shown different, say, nitrogen rates in, whatever crop might be. And then looking at the chlorophyll meter, you can see we've got grain yield, chlorophyll, and leaf nitrogen, and these things are all relatively related based on the actual fertilizer rate. So the idea, again, is for this to provide some indication of if you're sufficient or if you're deficient. There's some devil in those details, but for the purposes today, we'll skip over that. So another possibility is using a NDVI, Normalized Difference Vegetation Index. So this is technology that's used quite commonly for a lot of different things. There's a lot of satellite kind of high-level landscape or regional scale analysis based on kind of NDVI. There's lots of satellites that are outfitted with these, and they look at the ratio of essentially near versus visible wavelength. Here's the equation. It's near infrared minus visible over, it's the ratio of that versus near plus visible. Trimble sells these little handheld units. Are about $500, and you can walk through a crop and take readings. Again, it gives you a value of 0 to 100. One of the kind of the Achilles heels of both these and the chlorophyll meters are that to make them very useful, you need a nitrogen rich strip. and you're supposed to calibrate it based on that. So different varieties and different hybrids of different, environments will provide overall different absolute values. And so you need to essentially calibrate it um, with a kind of strip where we will assume that nitrogen or it's typically nitrogen is not the most limiting nutrient, that it's not a limiting nutrient. And so anyways, there's kind of devil in the details, but some people are using these and find them very useful for overall management. Okay. We can take technology like that and we can mount them on, say, fertilizer booms, and actually, this is technology that came out of Oklahoma State quite a while ago, and thinking about using this as kind of a variable rate on the go applicator. And so how this works, this is an example where you've got, say, nitrogen fertilizer liquid in this tank, and then you've got these green seekers on each, say, mounted, and then this machine will pass through the corn row, and it will look at, kind of give you a real-time NDVI reading. And then when it does that, it will either increase or decrease the flow rate individually on each one of these drop tubes. And you essentially are fertilizing in season corn, and provides, the opportunity to really tighten your nitrogen management and provide the nitrogen that the crop needs, not too little, not too much. So this is kind of the technology that might be that's out there. We don't see a lot of this in Ohio, but there's there's certainly people that do use it, and trying to kind of, there's, again, getting your system set up and running and having confidence in it. There's a lot of steep learning curves with that. So and then finally, just remote sensing, and we'll talk about this later in the semester, and kind of, we have essentially a precision ag week that we'll deal with at the end. But just to give you some idea, of course, there's a lot of, this is a field that's really exploded the last, say, 10 years with, smartphones and iPads, and just how cheap a lot of kind of the visualization equipment and as computer technology has expanded. So we can think about, using remote sense data that's coming from a satellite imagery, an airplane, or even like a little drone that might be flown over a field. There's always trade-offs versus cost and resolution. Satellite imagery is by far the most affordable, but it's also the lowest resolution. Flying an airplane over a field, you get good resolution, but relatively expensive. And thinking about a drone, you can get very high resolution information from a drone, but that drone's typically flown so low that it's hard to capture lots and lots of acres with the drone. They're limited by battery, the battery power, et cetera, et cetera. So flying something via an airplane, you can capture, tens of thousands of acres in a day. That's just absolutely not realistic to do with a single drone, right? So there's trade-offs, again, in terms of information you can capture and the cost of that information. But we use remote sensing to think about nutrients, about water, about pest outbreaks. You get these really pretty pictures that are heat maps, and make inferences based on that. We can use things like visible light, infrared, multispectral bands, thermal images. There's a lot of different technology in terms of measuring canopy reflectance, and the different wavelengths that you put together to kind of construct an index or whatever. There's some of this is, there's very well established technology, and there's approaches, and there's kind of new and emerging things. So this is a kind of a very exciting area in terms of the potential it has to play in agriculture, and how we can get information and scout fields and find problem areas and, et cetera, et cetera. So a lot more to say about this, and we'll save that for later in the semester. I just want to end by talking about, what to do with a number. So all of these things, we talked a little bit about visual symptoms, but anytime we send a sample to a lab or use, say, a Petiole nitrate meter and get a number, or find an NDVI value, like these are all numbers that, generate. And that's a good thing because it's an objective, repeatable, reproducible thing. But the big question is like, how do we actually interpret that number, and what does that number mean? How do we know what is a good number versus a high number, a low number, et cetera, et cetera? Well, and this, I was alluding to this a little bit earlier, but like this is one of the challenges of, and one of the things I want to really kind of drive home the point of this semester is that we should believe typically in the power of generating a number and having that number be repeatable, assuming our methodology is set and it's a valid approach. But what that number actually means and how we manage it, that is a whole second set of questions, right? So knowing what is a good number versus a low number versus a high number is a much larger step in terms of the information that's required to generate a recommendation, essentially. And so a lot of times these published book values might come from land-grant university, or it might come from like industry reports, or, like soil testing labs that say, well, we analyzed, over the last 10 years, we analyzed, 100,000 turf samples, and here are the, here's the distribution of those values. Well, if you've got 100,000 turf samples, you've probably got a pretty good distribution, a pretty good idea of what a typical value might be, what might be on the low side, what might be on the high side, right? So these are the ways that we do it. A take-home point is that, a lot of times you can get a book value, and that book value might not be very good, right? It might be old. It might be used with varieties that were, maybe some of that work was done 30 years ago, or maybe it was done, say, in another state or another region. So it's, I'm not trying to say none of this means anything, but we have to be very kind of skeptical and inquisitive about where these reference values are coming from and what they mean. And so just a word of caution, again, we'll talk more about this as the semester progresses, okay? So kind of from a practical perspective, thinking about different recommendations that we have for field crops, vegetable crops, fruit crops. We can talk more about this as we kind of get into some of these nutrients, but there's, of course, Ohio State has made recommendations and provided context and done work in terms of determining recommendations. But a lot of people are kind of shocked to see what work has actually gone into like something that you would think would be very widespread. Like what is I'm not picking on an industry, but like just imagine like you'd think that we have really good numbers for cabbage production in Ohio. Well, I'm not saying that we don't. I don't really know the particulars of those, but you might be surprised to find that like values for a particular crop or whatever there might not have ever been any cabbage fertility work that has ever happened in Ohio from an Ohio State perspective. Growers are constantly experimenting, and they might have industry reps or consultants that work with them and provide recommendations themselves, but kind of having that like really validated and gone through the quote unquote scientific process might not have happened because there's just a huge range of things that are grown in this state, right? So maybe that was borrowed from work that was done in Michigan or work that was done in Kentucky, or who knows. So that's not a real world example. I don't really know anything about where the recommendations for cabbage production came from, but just I'm just saying it as an example. You take a crop like corn or soybeans, and well, then we have a better idea because almost every state in the Midwest with, so many acres dedicated to that has done fertility work on that. So part of that is about the value of that industry, the number of acres that that particular crop might cover, et cetera, et cetera. All right, so again, we can talk more about that, as the semester progresses, but just to give you some heads up. Finally, I'll just say that, all of these kind of reference values have been compiled. This is really kind of the source for plant tissue analysis values. It deals with agronomic, horticultural, landscape crops, trees, and everything. So the whole kind of compilation of reference values that has gone into this. Again, some of these values are very good. Some of them might be a little bit squishy. Some of them might be place dependent. It's true, say, in North Carolina, but maybe not so much in Ohio. If you've looked up loblolly pine nutrient values, we'd probably be guaranteed that information, say, might have come from North Carolina and not Ohio, right? And so anyways, this is a nice book just to be aware of. It's an expensive book, but you can always, if you've got some crazy thing that you're looking at, like what are the range values for hops? Well, you're going to find it in this book. Okay, so that's again an overview of tissue analysis, tissue testing, and thinking through some of these issues. So we'll end here. And we're going to move on next week to looking at getting into some of the nutrients and then into acidity specifically, talking about soil acidity.